All right, everyone. Hello and welcome to our Midday Science Cafe. We were regaling you with the wonderful Havana parody nanobot. And yes, I can definitely put it in the chat in a little bit so that you guys can experience it yourself. The YouTube video is fantastic. And they have a lot of other fun science parodies as well. So I'll let you guys peruse that on your own time. Hello, everyone. Thank you so much for joining our October Midday Science Cafe in honor of National Nano Day, which was on October 9th. We are so excited to have you for nanotechnology, tiny science with a big impact, with Professor Grigory Tikhomirov from UC Berkeley and Dr. Emma Vargo from Berkeley Lab. Just a few things before we get started. You can find closed captioning for the Midday Science Cafe in Zoom. Either it popped up automatically or it's in the three dots under More, but feel free to open those up and follow us in text. This is being recorded and we will post the video to the Science at Cal YouTube channel and the Berkeley Lab YouTube channel, and we'll share that link with you all via email once it's posted. My name is Michelle Rodriguez, and I'm so happy to be with you all today. I'm Science at Cal's interim public engagement specialist, while our executive director, Dee, who you normally see on this screen, is out on maternity leave. As we normally do with our Midday Science Cafes and all of our programming, we're going to start today with a land acknowledgement. We acknowledge that Berkeley sits on the Huchun territory, the ancestral and unceded land of the Ohlone people, the successors of the historic and sovereign Verona Band of Alameda County. This land was and continues to be of great importance to the Ohlone tribe and other familial descendants of the Verona Band, and every member of the Berkeley community has and continues to benefit from the use and occupation of this land since the institution's founding in 1868. By offering this land acknowledgement, the Berkeley community not only recognizes the history of the land on which we stand, but also recognizes that the Ohlone people are alive and flourishing members of the Berkeley and broader Bay Area communities today. Thank you for allowing us this time to share that land acknowledgement. In 2008, Science at Cal was envisioned as a unifying effort to raise public awareness, understanding, and appreciation of scientific research at Berkeley. To realize this vision, we engaged the vast Berkeley science, technology, engineering, and mathematics community to foster creative collaborations among campus and community-based groups who share our commitment to equity and inclusion in STEM education and careers. Science at Cal connects researchers with diverse community groups of all ages and backgrounds for science engagement and learning. Accessibility, inclusiveness, creativity, and innovation are hallmarks of Science at Cal events, which reach tens of thousands of people annually. Throughout the year, Science at Cal presents ongoing free outreach programs on and off campus, helps promote related efforts, and creates new programs and initiatives at Berkeley and in the community. This broad scope of activities is made possible by Science at Cal's dynamic network of campus alliances and valuable community partnerships. And before I hand things over to Berkeley Lab, who is one of our valuable partnerships, to say hello, I want to remind you all that these events are always really interactive and we expect a ton of Q&A. So you can add your questions into the chat or in the Q&A box. You don't need to put them in both, and we'll get to as many questions as we can. I'm going to hand things over to Jocelyn Delgado with Berkeley Lab.
Thanks, Michelle. My name is Jocelyn Delgado, and I am the Senior Administrator of Community Relations at Lawrence Berkeley National Laboratory. For those who aren't familiar, Berkeley Lab is one of 17 U.S. Department of Energy national laboratories across the country. We're supported by DOE's Office of Science and managed by the University of California. All of the research we conduct is unclassified, and since our founding in 1931 by a UC Berkeley physics professor named Ernest Orlando Lawrence, we've been dedicated to advancing the scope of human knowledge and seeking answers to some of the greatest problems facing humankind. Today, Berkeley Lab employees work together to develop meaningful scientific solutions, to the world's most intractable energy and environmental challenges, help train the next generation of scientists and engineers, and ensure that those things happen in a manner that benefits everyone. Our main campus is located in the Berkeley Hills, and our close ties to the UC system creates a unique and synergistic environment for scientific discovery. Many of the lab's researchers are affiliated with one of the UC campuses as students, postdocs, and professors with joint appointments at the lab. We're fortunate to have an especially close relationship with UC Berkeley, and our institutions have joined forces to advance science across many frontiers. One of the main motivations for creating our Midday Science Cafe series is to share with you examples of compelling and complementary scientific research from both of our institutions. We hope you enjoyed today's presentations on nanoscience. And now it's my pleasure to invite our first speaker to the screen, Dr. Emma Vargo. Emma is a postdoc at the Center of High Precision Patterning Science. She received her PhD from UC Berkeley in May of 2023 and moved uphill to work at Berkeley Labs Molecular Foundry in June. Her graduate research focused on self-assembly of polymers and nanoparticles for applications of packaging, optical devices, and energy storage. As a postdoc, she's working on using polymer self-assembly to heal errors in microelectronic devices. Emma was originally drawn to research self-assembly because of its many parallels to biology, where amazingly complicated structures are encoded into relatively simple building blocks. She has found that combining design rules learned from nature with our rich library of man-made materials leads to exciting results. And outside of the lab, Emma enjoys knitting, sewing, baking, and watching her plants self-assemble into new leaves, which I also enjoy. Emma, over to you.
All right, thank you so much for that introduction. Hi everyone, thank you for being here, and I'm very excited to share my work with you. Today I want to share an example of how nanoscience and self-assembly can help us improve simple everyday materials. Specifically, I'm interested in exploring how we can use self-assembly to engineer better packaging materials. But before we dive in, I want to quickly define what we mean by nanoscience, nanoengineering, or nanomaterials. The prefix nano is the label for a specific size range, the nanometer. As humans, we're pretty comfortable with sizes from kilometers through millimeters, or maybe a little bit smaller. But nanomaterials are remarkably small, many thousands of times smaller than a human hair or a speck of dust. As one comparison, if you shrunk the Earth and everything on it to the size of a tennis ball, then all the tennis balls would be the size of a typical nanoparticle. Both of those are 100 million-fold shrinking in terms of size. Another rule of thumb is that nanomaterials are larger than atoms, but smaller than cells. On this plot, every step to the left is a 10-time decrease in size. So we see that a salt crystal is about 100 times smaller than a tennis ball, and a cell is about 100 times smaller than a salt crystal. Then between 100 and 10,000 times smaller than that brings us to the world of nanomaterials. On the top, we see biological nanomaterials, including proteins and DNA. And on the bottom, we see many different types of man-made nanoparticles. Given the huge variety of nanomaterials, both natural and man-made, there's a lot of fascinating science to be done around this size range. You may have seen in the news that quantum dots received this year's Nobel Prize in Chemistry. Quantum dots are one type of nanoparticle, where their special properties are a direct result of their tiny size. Each of the beautiful colors in the top picture comes from slightly different sizes of quantum dots. On the previous slide, you saw how many biological materials, like proteins, viruses, and DNA, are nanomaterials. And so if we want to design materials to mimic or interact with biology, we use nanoscale materials. These images show my own favorite nanoscale phenomenon, which is structural color. The blue butterfly, a morpho, has wings made out of a brown material. However, the material is arranged into a specific nanostructure, which almost looks like rows of pine trees. This nanostructure acts kind of like a prism to separate out different colors of light and reflects a beautiful blue color back to our eyes. Finally, if we think about building materials from the bottom up, by engineering the positions of individual atoms, we can create incredibly precise nanomaterials. These microscope images show human-designed stars and smiley faces made out of DNA. This approach to engineering is what Greg will describe in his talk. And so with that context in mind, today I'll talk about how we could use nanomaterials to design better packaging. This is work that I performed while I was a graduate student at UC Berkeley. Packaging protects sensitive, perishable, or reactive materials from the outside environment. We're able to ship packaged goods all around the world so people have access to more variety in food, medicine, and other consumables than ever before. This map shows how the United States in particular imports its food from essentially every corner of the world. Modern packaging is very reliable. When we eat packaged food or drink bottled water, we can be almost certain that it's safe. The basic job of packaging is pretty simple. We want to exclude things like dirt and bacteria, and we also want to exclude molecules like oxygen and water vapor, because they can cause uncontrolled chemical changes. If you've ever seen a sliced apple turn brown, you've watched how oxygen in the air can degrade food relatively quickly. In this example, the skin of the apple serves as its own packaging, but not all foods have built-in packaging like this. In this talk, I'm mostly using examples of food packaging, but the same ideas apply to medical supplies and electronic devices. In order for a material to behave reliably, we need to be able to control what's in its environment. The history of packaging shows us how both our science and our priorities have changed over a few decades. The first human-made packaging materials were glass and metal. These are dense materials with very tightly packed atoms, so they are very effective barriers against oxygen and water molecules. We still use glass and metal to package food when we want it to be safe for a very long time. However, glass and metal are heavy, and so transporting this packaging around the world is energy intensive. A second generation of packaging materials are based on plastic, which is cheap and lightweight. Plastic is pretty sturdy, but it's not as dense as metals or glasses, and so it's not as good of a barrier. In this case, we want to combine the density of metals with the lightweight and durability of plastics. A classic trick of material science, when we want to combine the beneficial properties of two materials, is to arrange them into a composite. There are many different ways to do this, including simply stacking thin layers of the different materials on top of each other. And so this is the approach used to design laminated packaging of films of metal and plastic layers. And once I learned about this type of packaging, I started seeing it absolutely everywhere. Generally, things like granola bars, candy, and chips will all be packaged in layer composite packaging. Now, great, we have packaging that is cheap, lightweight, and excellent at keeping food fresh. However, recycling materials like these is almost impossible. Even though the individual layer materials are recyclable, there's no good way to delaminate the layers. And so nearly all of this packaging material goes to landfills. Clearly, there are great opportunities for next-generation packaging materials to solve some of these problems. Especially around the city of Berkeley, we've seen a transition toward compostable packaging. This works great for many products, but we do have to sacrifice some barrier performance to make packaging compostable. And so as another option, my group and I have looked into making laminated packaging recyclable by harnessing self-assembly. Stacks of alternating hard and soft layers are a classic motif found in nature. Just like the layered packaging, alternating layers combine the advantage of two or more materials. Stacked layers create the strong, waterproof, and optically beautiful nacre that makes up pearls and shells. Similar layers are used to make the strong but lightweight armor of many insects, including this beetle. As an extra bonus of the layered structure, the beetle also gets beautiful iridescent structural color from the layers, similar to the morpho butterfly I showed before. Unfortunately for us, the building blocks for these systems are pretty complicated, and we can't easily adapt them to make packaging materials. However, we can design a simplified set of building blocks to make layered hard and soft structures. And to do this, we start with the idea that some materials simply do not like to touch. If you have a vinaigrette salad dressing in your fridge, you'll see how the oil and water parts will form two separate layers separated by a flat interface. If you shake the bottle and let it sit for a while, it reliably returns to the separated state. This is self-assembly. Scientifically speaking, oil and water are immiscible. This just means that they want to touch as little as possible. In the case of salad dressing, the liquid will reliably form two flat layers to minimize the contact area between oil and water. At the molecular level, molecules of oil and water will gradually rearrange without you having to do anything. Every time, the mixture will return to the same self-assembled state. This is annoying in salad dressing, but extremely powerful as an engineering tool. Along these lines, we can design new materials to harness immiscibility. Say I take small droplets of water and bond them to small droplets of oil. Now the system doesn't have the option of forming two big layers. The best it can do is to form alternating stripes of oil and water, so it's only touching along the bonded areas. And so simply, we've designed materials that will create long, flat layers automatically and reliably. And so you can see that we're getting close to our desired structure. To make our building blocks, we chemically bond water-like plastic molecules to oil-like plastic molecules. Plastic molecules are called polymers, and they're basically tiny floppy spaghetti noodles. If we give them freedom to rearrange, they'll flow over each other and ultimately form the structure we're looking for. A huge variety of chemistries are available, including the two-sided polymer chains that we need. And so at this point, we're halfway there. We have self-assembling nanoscale layers. However, we still need a way to make one of the plastic layers dense, like metal or glass. And once again, we can harness the power of self-assembly. We can buy metal or ceramic nanoparticles with a coating that matches one of the plastic layers. As the plastic molecules form strips, the nanoparticles pack closely into alternating layers. All we have to do for self-assembly to take place is to paint the mixture onto a base material and let it dry. Once dried, we have hundreds of almost perfect hard and soft layers. In this diagram here, I've left the middle steps of self-assembly blank because we don't know exactly what happens during the transition from disordered polymers to ordered layers. This really is the beauty of self-assembly. We don't need to know. We just have to combine the right ingredients and they'll take care of the rest. Once the mixture is dried, we're left with films on top of these base materials. This chip here is about 1 centimeter by 1 centimeter. I was very excited to see this beautiful blue structural color because structural color is a strong hint that we have ordered nanoscale organization. However, I want to look at the material directly to know how well it's assembled. One challenge of working with nanomaterials is that they're too small to see with light, even under a microscope. And so instead we have to use an electron microscope, which are kind of like big artificial eyes. Just like your eyes use lenses to focus light onto a retina, an electron microscope uses lenses to focus electrons onto an electron detecting screen. Using this screen, we can take photographs of extremely small objects. To prepare my films for electron microscopy, I use a very sharp knife to cut thin sections that are transparent to the electron beam. This allows me to see the cross-sectional structure of my self-assembled material. Getting to image these beautiful layers was one of the most rewarding and exciting moments of my time in grad school. The blend of polymers and nanoparticles worked just like we hoped and formed hundreds of flat layers. Here, the darkness maps out the density of the different areas, and so the darkest lines are the densest. If we zoom into any of these stripes, we see the individual tightly packed nanoparticles that make up the dense layers. The only constraint is that we can't let the mixture dry too quickly, otherwise it doesn't have time to self-assemble. The result is a patchy material with disorganized areas with and without nanoparticles inside. This is useful as a control material though, because we can separate out the effect of the ingredients from the effect of the ordered structure. Overall, this was a very exciting result. We don't need any glue to bond the layers because the polymer molecules tangle into a stable material. And this is one reason that this material is particularly recyclable. This is a photograph of the barrier test experiment, where I'm showing how well these materials can act as packaging. I like this photo because you can clearly see which material has an ordered structure based on its structural color, and which one was disordered based on its milky white color. We use the nanocomposite as a barrier between humid air and desiccant pellets. Desiccants are materials that suck water out of the air. By measuring the metal cans over time and weighing them, we can see which layers are acting as the best barriers. For the best packaging materials, we want this water transmission line to be as flat as possible. We compared the self-assembled layered material, shown in blue, to a disordered nanocomposite, shown in pink. We also used a standard plastic packaging as a control, shown in orange. As we hoped, the layered material has the least water transmission. This shows us that the organized structure is important, and that the nanoparticles alone aren't sufficient for improving the packaging performance. We really do need those long, flat layers. We also had some fun pulling on the material and making sure it's sturdy. This is a video of me squishing and stretching a film of pure nanocomposite between two pairs of tweezers. Finally, I tested the blend's recyclability by dissolving it in the same liquid it started out in. The layers dissolved almost immediately. After the nanocomposite disassembled back into its building blocks, I can paint the mixture onto a fresh base layer and recover the same layered structure. Theoretically, this cycle could be repeated indefinitely because there's no chemical transformations taking place during disassembly or reassembly. In comparison to current layered packaging, which needs to be unpeeled to be recycled, this nanocomposite could just be treated like a single type of plastic. Of course, these experiments are just the beginning. There's a long road between these results and being able to buy self-assembled packaging at the store someday. Here I've included some of the questions I still have, and you can probably think of even more. But despite all of these unknowns, I'm cautiously optimistic that self-assembled packaging materials could give us excellent performance of layered packaging without the recyclability problems. Before I end, I want to thank everyone who worked on this project with me, most importantly, my advisor, Professor Ting Xu, and other members of her research group. We're also very grateful to funding from the Departments of Energy and Defense. Finally, in the last minute, I want to recap the main points of this talk. As scientists, we're excited by nanomaterials because they can have new properties, behaviors, and interactions. Packaging materials must balance requirements of their barrier properties, lightweight, and recyclability. To combine the desirable properties of plastics and metals, we see that layered packaging materials are very common. However, they're essentially unrecyclable. By taking inspiration from salad dressing, we can engineer a blend of polymers and nanoparticles to automatically form layered structures. These nanocomposites perform well, and most critically, they can be easily disassembled and reassembled. Lots of open questions remain, but this is an exciting proof of concept. And with that, thank you so much for listening to my research presentation, and I look forward to answering some of your questions.
Thank you, Emma. That was a great presentation. And before we move on, we have a couple of questions. Our first question is, can the structural color of the self-assembled material be used for something?
Yeah, I think that's a great question, and something that I would absolutely be interested in studying more. The important thing about structural color is that the structure determines the color. And so if there was something to happen to change the structure at the nano level, even though we can't see the actual nanomaterials, we would be able to read that out as a change in the color. And so I think there's very cool potential applications where maybe the structure changes as a result of some contamination or the food going bad, and you would actually be able to see that the packaging has changed color, warning you that it's not safe to eat. And so I think it would be very cool to connect those two things.
Yeah, definitely. And what do you think is the most challenging part of working with nanomaterials?
Yeah, absolutely. Probably, as I mentioned, the fact that you can't see what you're working on. And so you go through, many weeks in the lab of, mixing solutions and heating things and cooling things. And you only know if you're successful at the very end when you look with a microscope. And so that makes it exciting, but it also means that sometimes you spend a lot of time that's ultimately wasted because you made a mistake early on that you couldn't catch.
Right. Okay, well, thank you so much for those thoughtful answers. Now let me hand things back over to Michelle to introduce our next speaker.
Thank you, Jocelyn, and thank you, Emma. That was fantastic. Really, really great. It is my current pleasure to introduce Greg to the screen, and Greg will be telling us all about his research. So Greg is an assistant professor in the Department of Electrical Engineering and Computer Sciences at UC Berkeley as of July 2021. Welcome to Berkeley. He has a long-standing dream to build systems approaching the complexity of life, motivated by the realization that incomprehensible natural complexity arises from comprehensible fundamental laws. Greg is interested both in understanding the principles required to build such systems, as well as in building practical devices using these principles. It is wonderful to hear your talk. I'm excited to hear it, and the Zoom screen is yours.
Okay, great. Thanks so much for the introduction. It's an honor to be here. I'm a new faculty at Berkeley, and I like to start my lectures with a quiz. So here's a short quiz for you all. What nanoscale device do all of us have in our pockets? And, I'm going to monitor Q&A or chat. Feel free to drop your answer, any crazy answer you think you have. Okay, I'm waiting. Okay, I'll give you some time. Feel free to add. But okay, one possible answer is a transistor. Yes, it is a nanoscale device. The thing with transistors, the smaller they are, the better they perform normally. And so most modern transistors are so tiny you cannot even see them. So there is a picture on the screen, of course, here, older transistor. And a funny fact about this building smaller transistors, the smaller they are, the bigger the factory you need to have to build them. So here's, for example, Intel factory in Arizona. And today, a new chip factory costs about $20 billion to build. So my goal for next maybe 10 years is to develop a new nanotechnology that would let us fit most of this Intel factory into a test tube. So, and if we look at the state-of-the-art transistors produced in this factory, we can see that each structure is a bit different. But if we zoom in on a pond cam in the parking lot near this factory, we can find an entirely different approach to nanofabrication, namely bottom-up self-assembly. A tiny bacterium about 1 micron in size can make thousands of complex, atomically precise nanostructures on its own. For example, every ATPase, and ATPase, of course, is a molecular motor that produces fuel for our cells. So every ATPase is made of exactly the same number of atoms. And you cannot say the same about transistors. And it's capable of producing complex behaviors at nanoscale. ATPase rotates at different speed depending on the voltage across the membrane. So the problem, however, that we cannot use this advanced nanotechnology from biology to build complex devices similar to computers. Due to evolutionary pathway to complexity, biological circuits are not designed in a modular way, amenable to engineering. For example, protein alpha crystalline, which is a major component of our eyes, of the lens of our eyes, it's also at the same time a key regulator for many metabolic processes. Trying to re-engineer this precisely tuned biological structures is extremely difficult. For example, if I want to change refractive index of a crystalline, I could inadvertently cause cancer. In contrast, with rational engineering approach, we can easily construct electrical circuits with scalable complexity from a simple set of composable elements, such as transistor, resistor, and capacitor. So fitting the factory into test tube was my job pitch that got me this job. And now I can tell you that my true dream, which is from middle school, was to create a new life form. And that came to me as a result of encountering the sickness and death in my family, like most of you. I thought that it would be nice to create a new life form, the form that would allow us to live longer and healthy and perhaps eliminate dying, but maybe upgrade our organs on the fly. And actually, the designs that nature gave us, some of them are really, really bad. For example, we eat and breathe through the same passageway, which is, of course, needless to say, a major choking hazard, killing thousands of Americans every year. So be careful right now if you're eating your lunch. And maybe I'll give you another quiz question to entertain you. Give me some other examples of bad designs in biology, in nature. I'm looking at the chat, Q&A, anything. Okay, I'll give you another one. For example, laryngeal nerve. That's something that I use right now to speak to you. It's a waste of 2 feet of wiring. So it goes all the way down from the brain to the aorta and then comes back. And as an engineer, I just want to nip it and reconnect it at the top. But of course, we evolved from fish. We cannot really do that. And in giraffes, it's maybe 14 feet of wasted wiring. And it's not only anatomical scale. If you go to nanoscale, rubisco, that's the most abundant enzyme in nature, because it's responsible for photosynthesis. So it converts CO2 into oxygen and sugar. And it is really bad at what it does. Typical enzymes convert maybe 1,000 molecules per second. So rubisco converts maybe 1 or 2 per second. Sometimes it grabs oxygen and burns it. So yeah, and nature decided, okay, that's good enough. Let's just scale up the production. We stop evolving this enzyme. And we ended up with this pretty bad enzyme. So the big question here is, can we combine the strengths of rational top-down engineering and bottom-up biomolecular assembly to build useful devices? I believe the answer is yes. But it's very hard, and I've searched for an answer for about 20 years. And then I encountered this field called DNA nanotechnology, and I sensed that that could be the way. So first, let me give you a brief refresher on the structure of DNA. It consists of a double helix of nucleotides that form a stack of complementary base pairs. Guanine pairs with cytosine, adenine with thymine, probably remember from your high school. So we know DNA mostly as a genetic molecule that largely determines who we are by regulating protein synthesis. In 1982, Nat Simon from NYU came up with a brilliant insight that DNA can be hijacked as a structural molecule to program assembly of new nanoscale shapes by base pairing rules that biology evolved for completely different purposes. A big breakthrough in DNA nanotech happened when Paul Rosamund came up with the idea of DNA origami. In DNA origami, one long DNA strand called the scaffold is folded by many short DNA strands called staples into any desired shape. So for example, here, the staple strand weaves this scaffold into a rectangle. Here you can see an animation of this process. It's really an amazing process that gives you a very high yield. And here are the snapshots of this process showing how a staple brings two different parts of the scaffold together by base pairing. In a similar way, DNA origami can be made into any arbitrary shape, like a smiley face, and short strands can be decorated with extensions to create any desired molecular patterns. For example, this map of Americas, which is just about 100 by 100 nanometers. So what's revolutionary here is that suddenly you endowed molecules with a programming language. By programming the right set of staple sequences, you can guide the scaffold to self-assemble into any shape you want. Go Bears. So single origami the size of Paul Rosamund's original smiley face is big enough to build just a single transistor by arranging, for example, carbon nanotubes on it. But if you want to build a functional circuit, then you need a much larger canvas to paint on. So I'd like to tell you about strategy to make a bigger canvas. First, we design this square origami, and in colors you have all the staples, and the black lines are the scaffold. And we look, let's say, we use atomic force microscopy to look at those nanoscale objects. And so Emma explained how TEM, transmission electron microscope, works by shooting electrons at your sample. So we here actually tap in our sample with a very sharp needle and scan it continuously through the sample. And that's how we can see nanoscale features of our self-assembled DNA nanostructures. So our strategy to make a larger DNA array was very simple. We take four square origamis, and we connect them into bigger square. And then we take four of those, make even bigger until we get to this 8 by 8 array of squares. And to see if it works, we try to self-assemble a nanoscale Mona Lisa. So we started with 64 unique tiles in 64 different test tubes. And we began assembling them in the groups of 4, and again in the groups of 4, till we get to this final structure. And so now let me explain how we connect these tiles. So on the first stage, we use this so-called extensions by 2 nucleotides and truncation on opposite tile by 2 nucleotides to create this sticky end interaction that is programmed by specific DNA kind of smart glue. On the second stage, we weaken it a little bit, we spread it out over a larger tile. And this way, we can get to our Mona Lisa in just 3 steps. And here's the final result. I hope you agree the AFM image looks quite consistent with the design. It's no Da Vinci, but we can make trillions and trillions of these nanostructures in parallel in one test tube. we created quite a few molecular paintings, including a life-size portrait of a bacterium and a bacterium-sized portrait of a rooster. And so now you might be wondering, this is all great, nice pictures, but what does it have to do with the real life? And so my goal for my research now at UC Berkeley is try to convert this engineering principles from DNA nanotechnology, from self-assembly and rational engineering to make useful devices. And so one useful device that I'll tell you, probably have only time to tell you today, is a biosensor for detecting viruses and bacteria. And let me tell you about viruses and bacteria. The way our immune system, our body senses and recognizes them is very interesting. It's actually typically multiple receptors on our immune cells, like macrophages, interact with multiple ligands on the surface of, let's say, bacteria drawn here, or viruses. Viruses have repeating patterns, spike proteins, we have many of them. And so how can our immune system recognize an invader and differentiate it from our own endogenous healthy cells precisely and decide, just kill the invader? Typically, it's a nanoscale arrangement of those surface ligands. We have pretty much the same ligands on our internal cells and invaders. Nature is not very good at designing brand new surface receptors and ligands, so it likes to recycle, but it arranges them in a little bit different nanoscale pattern. So here's an idea: use this DNA origami canvas in conjunction with field effect transistor to detect those pathogens. So typically, biofats have a random arrangement of these ligands. With our DNA origami, we can paint any pattern and we can attach any molecules to its surface, so we can precisely recognize our invader, our pathogen. And so we are now developing a way to detect them, and we use electrochemical way. Typically, you put this blue molecule here at the top. When it's close to an electrode and you apply a positive voltage to your electrode, you have a high current of electrons extracting them from this blue molecule. And when your target comes in this case, it's a DNA, this molecule moves out, and then you have low current, and you can detect this drop in current. And this is how you detect the specific sequence. So now, if we use this, let's say, just a smaller version of Mona Lisa, just 2x2 array, which is about 200 nanometers, and we attach many blue molecules here on electrodes. So it's a top view and a side view, and then let's say coronavirus comes in. So what happens is this DNA origami kind of wraps it around, eats it like Venus flytrap, it's a fly, and all of those blue molecules are now very far away from electrode surface. And so your change in your current will be huge compared to any existing sensors. Yes. So this is a way to detect bacteria or viruses, whatever you like. We want to make it agnostic to detect anything we want. And so this work has been done in collaboration with Lawrence Berkeley Lab, Ricardo Ruiz and Beihang Liu, and was led by talented postdoc in our lab, Lindu, and undergrad Liu. And so what we do, we pattern our entire wafer with patches that combine the small, quite large DNA origami arrays, and then we're going to have electronic detectors under those DNA origami, so we can detect multiple pathogens. And I think in the future, we can detect not only pathogens, but we can detect small molecules with high precision. And you can, I guess, imagine in the future that you don't go to your doctor maybe like once a year to do your blood test, your urine test. But we make those sensors so cheap that every day at home, you can just test your saliva or some other bodily fluids for presence of multiple, maybe hundreds of different biomarkers simultaneously, and current alternative would seem very medieval. So I guess that's all the time I have to talk about research. We do a lot of other different research from quantum dots to nanomachines to interface human brain with electronics. And I'm just happy that all these talented young people joined my lab and took a chance on the NUPI. And thank you for your attention.
Wonderful. Gregory, thank you so much for that really fascinating talk. I am just so inspired by thinking about how you're taking something proof of concept like the Mona Lisa into reality. I have a question for you sort of related to that. You'd mentioned being able to eventually one day being able to not go to the doctor's office as often, but rather being able to sense things at home. How far away do you think we really are from that application on a large scale?
Well, yeah, right now, many people, of course, have a very similar dream, and many people around the world working on this. FDA currently only approved a glucose sensor and maybe heart rate sensor. That's the only kind of wearable continuous monitoring sensors we have. But I think it would be, I think the sweat rate and some other like hormone sensing would be available to public and maybe in the next few years, not from our research group, but from other research groups. What we are trying to develop is universal mechanism to detect an arbitrary molecule, arbitrary protein. Yes, and that is probably like 10 years away.
How does that work if it's arbitrary? Does the at the end user, how do you make it so that it's arbitrary?
So the way to make it arbitrary is to use this DNA molecules called aptamers. And aptamers is just regular DNA. You create a random library and then you perform so-called directed evolution. You select aptamers that selectively bind a molecule that you're interested in, for example, a protein, a virus, a cancer cell surface. And then you can take this aptamer and attach to a DNA origami mechanism. And there is a lot of engineering there, how to design a mechanism that can be converted, binding to which can be converted into electronic signal. And that is, aptamers you can evolve for anything you want, so it's arbitrary.
Wow, how neat. That's awesome. I would love to invite Jocelyn and Emma back to the Zoom screen, and then we can start our Q&A in earnest. We've got some questions for you both. I think we'll start with Jocelyn to ask Emma some questions, and then as we're answering questions, you audience out there, feel free to add more questions into the chat or the Q&A box, and we'll get to them and we'll have a fun conversation about nanotechnologies.
Yeah, of course. So Emma, this question is for you. So what are some of the biggest technical challenges to scaling up your research and making it a viable product?
Yeah, that's a great question. First would be availability of the material. So right now we're buying our polymers. It's a commercial product, but we're buying them in very small quantities because they're sort of research grade materials. They're relatively expensive. And so someone would have to figure out how to make high quality polymers like the ones I'm working with at a very large scale. And there are similar polymers that are being used in things like tennis shoes. So it's certainly possible, but that is just kind of a big engineering challenge. The second would be getting, folks like the FDA on board with having nanoparticles in contact with our food, because that's something that we just don't know that much about. And so there's more testing that would have to be done on that end. And then finally, just figuring out how the actual manufacturing process would look. Because right now, food packaging is produced kind of in this continuous way where we're just continuously producing rolls and rolls and rolls of packaging. And so we'd have to figure out some way of painting the self-assembling material continuously such that it has time to self-assemble before the assembly process or the manufacturing process is complete, which I think would be a very fun project to work on, but one that you'll need a ton of material to pinpoint exactly how to do that. So those are the three that I can think of.
Okay. And speaking of food packaging, do you think you can push this even further and make this packaging edible?
I think that's a great idea. I think the polymeric part, definitely. There's tons and tons of edible polymers. Really, a lot of the foods that we eat are polymers. The nanoparticles gives me a little more pause. because I'm not completely aware of edible nanoparticles, but I bet they're out there. And if you wanted to work on that, that could be very cool, where it would work great as packaging, but then, you know, your body can break it down at the end of its life.
And do you think there are any safety concerns when it comes to packaging of food using this material?
Yeah, that's something that I would want to be very mindful of. The thing about nanomaterials is it's a relatively new field. And so we just don't really know the effects on a lot of things. And so right now, the policy is to really err on the side of caution. And, you know, I use gloves when I'm handling these materials, and we work in kind of these contained chemical environments. But at the same time, we have things like sunscreen that include titanium dioxide nanoparticles that we've decided is safe for our skin. And so I think we can't know for sure, but there does seem to be good evidence that nanoparticles can be used in our skin or near our body and not cause any sort of harm. And so I think testing is needed, but it's not necessarily a deal breaker.
Yeah.
Greg, I have a question for you. You were just talking about the aptameters. How stable are the DNA nanostructures like the aptameters for the sensor domains, especially when it comes to like shelf life?
Yeah, good question. Typically, DNA is not very stable in vivo because our body is trained to recognize and chop it up. We have this nucleases that eat up DNA. When you attach it to a surface like a sensor surface, they become much more stable. And if let's say that stability is not enough, you can chemically modify some of the bases to be unrecognizable by enzymes. So that's actually another Nobel Prize this year for RNA vaccines. You know, the kind of full immune system change up with some bases in mRNA not to be recognized by enzymes, and that's how you make it more stable.
Thank you. Here's a question that we like to ask folks. Well, how did you become interested, Greg, in nanoscience? What was the draw for you?
Well, I alluded to that a little bit. I started with biology, actually. I was fascinated by complexity of biological systems. Like, I thought that it's amazing feat of engineering to be able to come to this just through stochastic evolution. But, you know, then around, you know, high school, you know, I told you I encountered some death and sickness in my family, like most of us. And I realized also from just studying biology, there are many bad designs, right? And, you know, could we maybe make ourselves healthy and then maybe immortal if you choose so? But and I went all the way down to molecular scale, I would say angstrom technology, and I realized that the tools of covalent chemistry are not sufficient to, let's say, assemble even simple cell. And so kind of found this medium in nanoscale where I think we have a lot of precision, a lot of control, and we can re-engineer these building blocks. And there is nano is where a lot of magic happens. It's a language of biology, and now it's everything. Almost all devices we make also nanoscale.
I love that. Thinking about nanoscales where the magic happens. It does seem like it. It seems like we've got some pretty like revolutionary ways to solve some of our problems, you know. Both Emma, you're thinking about like recyclability and thinking about reducing the waste that we have, and Greg, you've been researching work on like our human health and being able to understand that. Greg, do you have other, I saw on the slides, you know, that you had two other sort of babies that you have that you're working on. Do you want to describe maybe one other? We have some time if you'd like to share.
Yeah, I have so many babies. I mean, and by my babies, it's my students in the lab. This is my main babies, and every one of them has 3 babies usually because they have one very ambitious project that they're really excited about. And so, okay, I cannot really choose my favorite kid, but you know, like one of those maybe is we're trying to develop a sensor to sense voltage of the neurons. So my first grad student, Benjamin, he played hockey for MIT, and he got concussion, and he since then became interested in kind of debugging neural circuits of the brain. and he's developing this. And typically, to monitor brain activity, you have to drill a hole, you have to insert electrodes, so nasty stuff. You cannot do it with regular people. So can we do it non-invasively? And so he designed this DNA origami piston sensor that would insert into membrane of the neuron, and non-invasively using MRI, because magnetic field can go through the tissues unattenuated, unlike light, can measure, can tell you whether neuron fires or doesn't fire by having this DNA piston moving up and down, and some magnetic nanoparticle like iron oxide, gadolinium nanoparticles. So yeah, this is kind of molecular machine that we're designing. I'm excited about that, but we also have several other projects from nanobots that deliver vaccines to nanophotonic devices and so on. Yeah.
That's amazing. Graduate work is so, and lab work is just so diverse. It's just wonderful that you're supporting that in your lab. Jocelyn, you want to take some questions?
So our next question is posed from one of our attendees here. So he said only about 6 to 7% of plastics can be recycled in the US. Does your research suggest some remedies for the problems posed by various types or all forms of plastics? And this question is for Emma.
Yeah, that is a fantastic point, and I was kind of hoping someone would bring that up, because I think it points to just how thorny this issue is. And so as I mentioned just briefly, I think compostable plastics are an amazing field of material, and something that I think is probably a big part of the cure for our single-use plastic problem. But however, making something more compostable naturally makes it worse of a barrier. And so to make something compostable, you need water and oxygen and bacteria to be able to break that down. And that's exactly what we're trying to prevent with our food packaging. And so I think we need just sort of this multi-pronged approach where things like paper cups or to-go cups can be made out of compostable materials, because they don't need to last very long and they can break down quickly. But then things like our medical supplies, for example, where we really want to be sure that nothing is entering that packaging, can be wrapped up in things more like what I'm talking about with the layered self-assembly material, where that could be recycled at the end of its life rather than thrown away. But yeah, I think there's no one material that's going to solve the plastics problem, unfortunately. So we're all going to have to figure out where to apply these different solutions.
Definitely. So you mentioned something about composite nanomaterials. So the technology that you are studying can make different shapes, such as like maybe use that material into maybe water bottles or reusable cups, like you were saying. So is that the goal of the research that you're conducting?
Yeah, right now we're mostly formulating it as a coating. And so it's very similar to paint. So when you paint is a suspension of little colloids in a solvent, and that solvent evaporates and you're left with the paint coating. And my material works exactly the same way, where we can kind of smear it on or drop it on, and then the solvent evaporates and you're left with a uniform coating. And so anything you could paint, I think you could coat with this material. If we wanted to make things that were standalone cups, we might have to do a little more engineering to make it sturdier, because right now it's just kind of this floppy film. But yeah, you should be able to paint this on anything you wanted, essentially.
Oh, that's amazing. And this question is for both of you. So aside from your own research, what developments in the field of nanoscience that could revolutionize our future excite you the most? Greg, would you like to go first?
I'm happy to start, or if you want to go, Emma.
Go ahead.
Okay. Yeah, I think, as I mentioned, nanoscale is where magic happens. And, I'm particularly interested in combining the strengths of top-down rational engineering with bottom-up self-assembly, because, nature kind of self-assembled us from atoms, but they did so in a very convoluted way. So we cannot, it's basically an alien technology. We do not understand it. We understand a lot of it, but we still cannot build them in simple living cell, not talking about human being. So what I'm very excited about is trying to use our rational thinking to be able to engineer artificial systems approaching complexity of living systems. And also once we this will take longer than my lifetime. Maybe some of you can do that. But what I think we can do already now is to integrate living systems with electronics. Can we develop maybe a way to communicate our thoughts into electronics, or maybe have a memory in our brain that is connected? You don't have to go to school. You have the access to Wikipedia through your brain. So yeah, those are some examples.
Yeah, I think I have maybe a complementary but opposite viewpoint, where I work with synthetic materials, so man-made materials, solving kind of man-made problems. But whenever I can read something that is just diving into, how does this biological organism work? How does nacre self-assemble? How do bacteria self-assemble? It just fills me with so much like admiration and excitement and inspiration from the biological world, where that's not what I'm working on specifically, but it is just incredible that we have this inspiration all around us. We ourselves are self-assembled, as Greg mentioned. And so there's just so much to learn about and be excited about in the natural world.
There is, it's so neat to see this. You guys have such complementary focus, right? It's just so interesting to see how nanoscale and nanotechnologies are so varied. Before this talk, I always thought of them as just like one aspect of the world, but it really has sort of there are many aspects within this like field of work, which is really neat. Greg, this question is for you. Are we expecting DNA nanotechnology field to follow the exponential growth trajectory observed in Moore's law? And not knowing much about this field, maybe if you could also describe Moore's law for some of our folks in the audience, that would be super helpful.
Yeah, that's a great question, and that's a great observation. So we have some specialists in electronics here today in the audience. Yeah, so the Moore's law is kind of increasing the density of transistors every couple of years. We have twice the density, and that kind of has driven all this revolution in computer science and AI. You hear about ChatGPT-3. It's because of this Moore's law. We also have much cheaper because you make transistors much smaller. They're more energy efficient. We can pack more transistors. So this DNA nanotech, the field started maybe 20 years ago, and now we have 300 groups around the world. And I mean, it's not a commercial technology, which is, Moore's law was driven by profit a lot, and there is a lot of trillions and trillions of funding around the world that was poured into the making technology better and better. DNA nanotech hasn't yet delivered, any killer apps, and it's a relatively new field, and it's yet to see what's going to happen. And also we kind of have an inverse. We have a fixed feature size, so we have about 2 nanometer resolution. And what our challenge has been so far is trying to scale up to increase the size of, let's say, uniquely addressable complex structures. So monoliths is one example where we managed to make very big structures. And I think now DNA nanotech is already at the stage where we can integrate it with top-down lithography, top-down fabrication approaches to build hybrid devices. I don't think we need to make structures even bigger. But, you heard about biotech, CRISPR-Cas9, Jennifer Doudna got Nobel Prize. And, one of the Moore's law-like tendencies in DNA is the cost of DNA is decreasing faster than the Moore's law, much faster. So every couple of years, we have reduction in the cost of arbitrary DNA sequence. So let's say we can edit our genes or try out expression of a new protein. And this is much faster than exponential.
Wow. Thank you for the explanation and for your response. I appreciate that. This sort of makes me think of where we're going in the future and like ethical are there ethical considerations? This question, I guess, is for both of you, but maybe Greg, you can answer first. Like, are there ethical considerations that you have to be wary of in nanoscale, like as you would in like AI computing or in CRISPR-Cas9 and thinking about like editing a genome? Are there similar like parallels?
Yes, so we're not really modifying genes of humans. We just use DNA as a smart polymer. So Emma was talking about using polymers to organize this layered structures, layered materials. We do exactly the same. We basically just use DNA as a smart glue to make our nanostructures, some interesting structures that have interesting physics, so we can use it in the devices. So we're not editing DNA of any people. But we do want to actually develop a gene delivery technology based on DNA, and there are yes, ethical concerns like everywhere in any kind of science. For now, I think the ability to cure people from cancer or do gene therapy that definitely overweigh and motivate us to develop this technology.
Thank you. Emma, how about in your field?
Yeah. So just to piggyback on that, I think there probably aren't the sort of very thorny ethical issues like you mentioned with gene editing or artificial intelligence. But I think we have an opportunity, if we're designing these new materials from scratch, to really, slow down and think about the whole life cycle of the material. Where are the raw ingredients coming from? Where does it go after we're done working with it? Because a lot of the materials that we are using currently were really produced in this very, very fast, breakneck cycle of discovery and innovation and application. And so, us working in the lab, I think we need to think about, where does this come from? Where does it go? How is this going to affect the world after I've finished, writing my paper, getting my PhD? And so that is something that probably all scientists are trying to keep in mind currently.
Thank you. It's so like, I don't know, thinking about science for good and science for having to think about, what we're doing is important. So I appreciate your guys' thinking about that. Both of you mentioned too, so you can't really see your work, as both of you sort of mentioned, but you're using technologies to be able to visualize, like you can see the Mona Lisa, or you can see the two different microscopy technologies. Are there other technologies or specific tools commonly used in nanoscience research that are essential for your work? This, I guess, is a question for both of you.
Yeah, I can start off. So I use if you look into the Berkeley Hills, you can see sort of this dome, and that is the synchrotron at Berkeley National Lab. And the synchrotron is an X-ray source and accelerator, and so it's organized sort of like a sun, where there's a core and then lots of different rays of X-rays shooting out of it. And similarly to how I said we can't use light to look at nanostructures, X-rays are actually smaller versions of light. And so that's light that we can use to look at nanostructures. Those are photons with the right size wavelength to look at nanostructures. And so scattering is kind of the overall class of characterization techniques involving using X-rays to probe nanostructures. It's a little bit harder than doing electron microscopy, because when we do electron microscopy, we get an image that's very similar to what we would see with our eyes, and we can interpret it very easily. Versus when we use X-ray scattering, it's very powerful, but also our output is just kind of a bunch of math, and we have to take the math and convert it and fit it and model it in order to figure out what we're looking at. And so that is a very powerful but very difficult way of learning more about our nanomaterials.
Yeah, just to add to what Emma said, we also use light, and we use visible light and not X-ray. Of course, visible light, you can imagine, very hard to use to look at the nanoscale, the wavelengths, on average, let's say 400, 500 nanometers. But it's sufficient to see some microstructures. And for example, when we develop our DNA nanorobot and we put it inside the cell, we put some fluorescent molecule, kind of fluorescent tags, and we look at those cells that are several tens of microns, see if the nanobot got inside the cell by looking at the fluorescent microscopy images, and their resolution is plenty. And in reality, you can actually use a fluorescent microscope to see nanoscale features. There are some tricks to do that. You can do 5 nanometer resolution with a 500 nanometer laser, and if you're interested in that, you can check out some papers on DNA paint. We also use electron diffraction and all other techniques so that in our lab we have TEM, AFM, fluorescent microscope, conductivity measurements. It's an array of tools that we go for many tools actually to Lawrence Berkeley lab, because some of the tools are much better than on Berkeley campus, and they let us use them for free because they're nice.
And the next question is for Emma. So this is a little bit of a technical question. One of our audience member wants to know, could you reiterate on what is the underlying mechanism of such layered nanomaterials for preventing water transmission better compared with the single polymer components?
Yes, absolutely. And so you can think about these layers as forming kind of roads for water vapor or oxygen molecules to travel through. And so you can think about, if you have just short little layers, there's going to be lots of gaps between them, and the water molecule is going to be able to tunnel up through it. Or if you have clusters of nanoparticles that are sort of disorganized, it's going to be able to find a path. But if you have these long continuous layers, it's going to have to go, all the way this way, all the way this way, all the way this way to get through your material. And so basically you're just trying to make it so annoying for that water vapor molecule to get through the material that it ends up just sort of statistically turning around and coming back out the other side without going through. And so you're just really trying to make it travel as far as possible. And the reason we can't just have the whole material being densely packed with all these nanoparticles is because then it doesn't have the really good mechanical stability. Because the reason that we're able to tug and stretch on the material I made is because of the polymer layers that are all tangled up. And so we need those polymer layers to give it the mechanical stability, and then the nanoparticle layers to give it the really good barrier properties. And that's the principle of the composite.
Okay. And then we have one follow-up question regarding the comment we made about the FDA. Has your lab worked with FDA to determine the regulatory pathway for food packaging products?
Yeah, so I am no longer in that lab. I've graduated, but my advisor, Ting Xu, I think has a friend at the FDA. And so she was hoping to reach out to them at some point and talk about, what would that pipeline look like? Even if it was going to be, a 10-year process, where would we start? And so, yeah, that's absolutely something we're thinking about.
Are you continuing any affiliation now that you've graduated from the program?
Yeah, I trained the younger students, and so we have some really great students taking over the project. And of course, we're still in touch, and we can still, they can update me, and I can, answer questions if they have them. And because, Berkeley National Lab is so close to UC Berkeley, I'm assuming we're just going to cross paths quite a lot. But I'm excited to see where the project goes.
Me too. Let's see, we have one more question. This question is for both of you. So we had a little bit of a fun comment. One of our audience members says, I'm a mad scientist, and in my quest to take over the world, I like to create nano machines. How far are we from having nano-sized devices that use energy to achieve this?
Well, welcome, fellow mad scientists. I am one too, and I would like to know the answer to that question as well. And we are trying now to pump energy in various ways into our nanomachines. And yes, light, chemical. I mean, we are actually made of nanomachines that use light, and ATP, right? That I refer to, that power our nanomachines, like ion channels, bacterial cilia, bacterial tails. And as I mentioned, it's very hard to engineer a very simple machine that nature can do it very well. And once we achieve some progress on this, I will contact you so we can take over the world together. Well, what we need is some funding. And $10 million would be enough for maybe first 2 years.
Emma, anything to add to that?
I think Greg has it taken care of. I'll stick with my packaging materials.
That's great. What a great question to end on. And someone just dropped into the chat an announcement from the National Nanotechnology Coordination Office to folks interested in nanotechnology. There is free registration for something called Enabling the Nanotechnology Revolution, celebrating the 20th anniversary of the 21st Century Nanotechnology Research and Development Act, which is on March 4th. This must be a typo. 2024 is my guess, unless we're also developing time machines and we can go back in time, which would also be fun. Yeah, 2024. Awesome. So thank you so much for dropping that into the chat for folks who might be interested. There's a link there as well. So thanks for that. I am going to thank our panelists so much for such a wonderful discussion about such a tiny subject, but it looks huge, and I love this. I'm going to leave this with the idea that there's like magic in the nanoscale and in nanotechnology, and maybe we're all a little bit mad scientists out there in ourselves. I'm going to share my screen one more time because I have a survey link for you all to tell us how we're doing. So let us know. And also want to make a plug for next month's Midday Science Cafe, which is always the third Thursday of the month. And it will be all about human health. So stay tuned for information about that. We've got some great speakers lined up for that too. And just thank you all so much. I don't know, Jocelyn, do you have anything to add about while we wrap up our Midday Science Cafe this month?
No, I'd just like to thank Emma and Greg one more time for all their both of their great presentations. I loved seeing the art and the science come together to explain something really beautiful. And thank you to our audience for showing up and asking such great questions. And as always, if you'd like to stay up to date on research coming out of our institutions, you can visit Science at Cal at science@cal.berkeley.edu and Berkeley Lab at lbl.gov.
Thank you so
Thank
much.
you all so much.
Thank you.
Bye, everyone.